Hello viewers, welcome to my channel of Biotechnology Tutorial and Research Lab. I am Associate Professor Dr. Sharif Hussain. Today I am discussing my lecture topic is branches and implementation of biotechnology in different fields in, like in animals, plants, environmental recycling and industrial product manufacture and others. First, I can differentiate of biology and biotechnology. It means, what is biology? Biology is study, learning and teaching of bio means life. Study of life. This is called biology. Biotechnology, study of techniques or technology related to life. This is the difference, biology and biotechnology. So, if you can say what is the difference between biology and biotechnology, easily we can say biology is the study of bio means life. This is in a short we can say biology. And biotechnology, study of techniques or technology related to life, environment, industry, engineering, medical, biomedical, etc. to comfort human life, having research and innovation, and finally practical implementation to give benefit in human and social life. This is called the biotechnology in details. So application of biotechnology or branches of biotechnology, different different branch of biotechnology. So we can see here, this is the like plant biotechnology, animal biotechnology, medical biotechnology, sometimes we say it human biotechnology, then environmental biotechnology, industrial biotechnology, engineering biotechnology, biomedical engineering biotechnology, pharmaceutical biotechnology, bio nanotechnology, microbial biotechnology. This is the branches of biotechnology or different types of biotechnology. So actually biotechnology meaning is broad, very broad. So let's go to discuss the of different types of scientific data in different field, like plant biotechnology, fruits and productions and quality development, like different types of, we will discuss today outline of scientific presentations for plant biotechnology. Already two PhD students completed these two projects. Micropropagation project for tissue culture biotechnology, one MSc student completed this project and one PhD student also completed now. Seedless vegetable productions, one PhD student completed. And flower enlargement and induction project. One MSc student completed this project. Then environmental biotechnology, biofuel productions and engine emissions test, biodiesel, bioethanol from waste, different type of waste. This is industrial biomaterials and bioplastic productions. Also biofuel productions. This industrial biotechnology belongs to industrial biotechnology. So environmental pollutions control also this is belongs to environmental biotechnology. Environmental waste management also environmental biotechnology. So also others biotechnology, like now you can see uh, engineering biotechnology related to engineering equipment developments, different types of uh, like chemical engineering biotechnology, application of biotechnology to chemical engineering. The biomedical engineering biotechnology, like cell culture and tissue engineering, and also it's called biomaterials productions. Pharmaceutical biotechnology, biomedicine productions, biomedicine, and for nanomedicine, nanobiomedicine productions, and also others. This is called others, the uh, medicinal plant, extraction of medicinal plants and produce different types of natural products, natural biomedicine. This belongs to pharmaceutical biotechnology, bio-nanotechnology. Then nano-size productions of biomaterials, different types of biosources. The microbial biotechnology, using the bacteria, virus, algae, different types of microbes. An application of it in different field, especially in the industry. This is called microbial biotechnology. So this is all about the topic today we'll discuss here. We can see here now. This is actually, this belongs to plant biotechnology. Export sunlight effect in winter and summer by cutting branches. Actually, here this is very good topic. This research I did in Japan. We produced the dwarf plant. This is dwarf plant. This is called the long plant or higher plant, very high plant. So we use the sunlight penetration to penetrate more sunlight here. Then plant get more energy and also produces more photosynthesis. After getting more for photosynthesis, it produces more sugar and plant can reserve more energy. By this way we can see the result, and this is the control long plant. We did not me and my professor, we did not cut this plant, so we can see the results. Flower, more initiation flower, we got more flower initiated in summer. In winter, in summer is less flower, but it is called the very effective dwarf size. And winter also very weak. This is very weak, this is very strong. Also you can see the plant, weak plant and strong plant. So you can see here dwarf plant, dwarf size, and then this is the this is for compact size, and this is the tall size, tall plants, tall and short. These two types of plants. So this is belongs to plant biotechnology. We use this to make the dwarf trees by environmental factor, sunlight. Then we can see here the color also developed. Different color, light factor. They got more light factor. Then color developed, and sweetness also developed. Then we can see here different types of test, nutrient test, nitrogen, potassium, phosphorus, calcium, magnesium. We tested and showed leaves and the fruits contains also total carbohydrate, fructose, glucose, sorbitol, then sucrose, starch, and they also different contents. You can see here. Then after that, just the shoot and the leaves. So you can see in the dwarf plants we got the higher carbohydrate than tall plants. Now we can see here SSC, soluble solid contents, with sugar contents and acid content in different years. The acid content when decreasing, the sugar content it was increasing in different years. You can see here 13 carbon determinations, which are 13 carbon, and artificial photosynthesis we produced here. Artificial photosynthesis. This is the fan, circulating the fan, and then here cover, plastic cover, and this is barium carbonate petri dish. The petri dish that barium carbonate. Artificial we produce the and artificial and show the 13 carbon monoxide fading in potted trees. So then we use filtered electric in a small pan for the air, and then we produce the artificial photosynthesis. Then we can see the results here. The treatment, leaves, shoot, and others. Roots, leaves we got the higher. This is the control. This is the treated. Bark rings and complete rings we got the higher carbon, 13 carbon in the treated branches only, then the control. Then you can see fruits developed also. Food developed more color. So the treated plants that control, some control. Then we did the analysis, abscisic acid by nanosite measurement, and gibberellic acid and cytokine isolation by the paper chromatography using tissue culture methods. We use this tissue culture method, analyze the abscisic acid analysis, gibberellic acid and cytokine. Then we can see here one result. It was higher in the treated branches than the control. This is all belongs to the plant biotechnology. Phloem and xylem stress. We did the xylem stress by the here in the cut razor blade in the plants, and then mid cut partially, mid cut 1 week, mid cut continuously. Then we can see the results. Different of color, food color developed, more color developed in the treated plant than control, because the treated plant they reserve more sugar. When you cut here, the photosynthesis could not go down from above. That's why reserved food was here carbohydrate reserved was reserved. That's why we got this carbohydrate used for the food developments. This then we can see this is the result. The other plant biotechnology food project, PhD student did this project. He completed effect of different growth regulators on the water apple fruits growth and development. He used this the control, and he used the GA3 treatment, gibberellic acid, and the earlier color developed and matured earlier 40. This is 14 days. Then here 3 weeks. This is the control, and this is the GA3 developed fruits are bigger and also color developed. 3 weeks fruits, we can compare between control and GA3 developed. GA3 50 ml per milligram per liter. So this is, we can see here, very good result. Fruit size also here, early maturity, then we can get here early maturity and color developed, more and here control, not yet color developed, but here 1 month, after 1 month treatment developed. So here different data also, so we got the higher fruit length, fruit color, the treated fruits that control. Then we measured the different types of TSS, means bricks content, total soluble solids, total sugars, flavonoid contents. You can see those contents, potassium contents, TSS, total sugar, flavonoid, total phenol contents, all the higher in the almost in the treated plants, treated fruits, then the controls. Here we can see effect of IAA. Before you used GA3 gibberellic acid, now indole acetic acid. This is the called organic plant hormone. So organic means, and then this is also same. After 3 weeks, the result IA treated fruits is very bigger than the control. You can see here the result. This effect of NAA, different types of NAA. We use the NAA, oxin, and we see the result in NAA. You see biomass content, pH, TSS, total sugar, so higher in different concentrations. And then at 10 milligram per liter shows the highest result. And also here you can see potassium content, total phenol, total flavonoid, reactive DPPS. This is the antioxidant. Also very good. You have seen here the highest antioxidant DPPS in the, this is called the Na10 milligram per liter. So this is the very good potassium content we got also here. There we can see different types of yield loss due to excess fruit dropping. Excess fruit drop during the experiment. Then in Malaysia, dropping rates of, for example, wax apple or water apple is more than 80%. So it causes the reduction of yield of jambu madu. Jambu madu means the, it is Malay language. Jambu madu means the water apple. So yield losses every year dropped, lot of excess food dropped, then we prevented dropping and we increased the loss of dropping. So, we prevented, and then gibberellic acid we can see here. Here we can see we decrease the we got the result and we decrease the fruit dropping. Here you can see now this is the gibberellic acid, the result of gibberellic acid. We use different types of the fruits contents, control, and also other more color developed. Then the control. Completed thesis of my PhD student. My PhD student did this work and completed. I supervised this student. This is another project. This was the beginning the fruits, different portions. This was from the Himalayas. This is the middle portions, and this is the distal portions. Distal, and there is three portions of the fruit, 3 parts of the fruits. So usually these parts of the fruits is not sweet. Middle one is more sweet. Middle one is middle or distal, and one is usually more sweet. That's why we did the sometimes distal and one, sometimes middle one is sweet, but for example one not more sweeter than middle portion and distal portion. So you can see the blended portion inside light shady. There is no light penetration here. Outside food more light penetrated, and then the outside food also. And this is the controls also for bigger size. Then the control. Then we can see here different types of the acid on TSS content in the foods. So you have proximants, middle and distillants. Middles usually got more. This is called what brings more sweetness than the distillant and the proximants. Here we can then bring fruits quality development by foreign states. Fig fruits, other fruits. You can see here we did the bark cutting or phloem stress. This is xylem, this is the phloems. We cut the phloem tissue and then to make the, it's called the restriction to calm down the photosynthesis. So in that case, this photosynthesis used for fruit development and fruit become more colorful and bigger than the control. You can see also cross X shape cutting, and then fruits is more bigger than control. This is micropower and other experiment plant tissue culture project. This is the plants isolated cell or tissue, then excised piece of leaves. This is the piece slice leaf slice, then we plant it here. This is the plants, and the isolated cell, then again plant it again leaves. So after this, from this planted one, we got the overview of the tissue culture process. This is called plant growth tissue culture. So you can see here different types of projects. Image of the project of tissue culture, class formations in the laboratory and the plant production in the laboratory. Image of from different scientist data. This is also from different scientist data. Our data also there. Here to different scientist data. You can see here project tissue culture in different types of plants. Here also we did the tissue culture in broccoli plants. Broccoli plants we got class formation. Broccoli plants in the laboratory, and then from these broccoli plants we did IABAP, and then we produced the plants, and then we planted it in the field and got broccoli. And this is also combination shoot tips and leaf from leaves, and we can see the class formations. This is the class formations. Then here we can see broccoli project in between. Then after that it can be transferred to the field broccoli plant, and then broccoli vegetable or fruits can be grown. This is somatic embryogenesis. Induction of embryogenic callus is achieved using a MS medium Russian source medium identification of broccoli. So this is the broccoli. This is the somatic embryogenesis. We use the embryogenic callus. We use the embryo, and then here from ornamental plants, nodal explants produced. Now you can see different types of shoots, preparation, number of roots, and other induction of callus formation, weights, and observations. Different type of data we can see in different concentrations, and we got the good result in the treated plant and control. Yeah, like the formation of pineapples from crowns. So this is the micropropagations. PhD student did this project. This is the crowns, cuttings in the crowns, and then grown the crowns in the bottle, and then after that this is on the beam, small beam, and then after that we can see the plants was grown. After the second culture, we can do the same culture, different types of media preparation, MS media, agar solutions, sucrose, electrolyte, microelectronic, vitamins, autoclaved. Then we made it autoclaves to 1 degree centigrade for 15 minutes. Then you see here image of different types of image. You can see. So also different types of the trees, small and bigger at different concentrations. Also different types of, here we can see second subculture from plantlet, second subculture, first subculture, second subculture from this culture. We got the plants, pineapple plants, and the data, BAP and A, different concentrations, and then you can see here the results better than the control. Research strips with my PhD student, my MSc student. We did the research strips. This MSc student, 2 students, and we visited Malaysia, and then I supervised this student, and we went to our place, mangrove plants, and this is medicinal plants. To collect the medicinal plants, we collected it. This is the University Research Center, and then students doing the research work. This is the 2 theses have been examined from UM and UPM, University of Malaya, UPM, Malaysia. At that time I was in the University of Hyde. I was teaching there, and then this is called the Yahoo Messenger. Skype virtual interview of 2 students. Attended the Viva of MS students from Hyde University as external examiner from University of Malaya by video call in Yahoo Messenger. You can see here. Then other experiment, seedless okra productions, injection methods, different types of methods, plant growth regulators, and then different types of zebratic acid we used, and AIA, different types of flower budding, stem injections, no more spray method, no polluted, environment, cost effective, significant. Seed reduces seed loss, seedless okra project. We produce seedless or seed, this is called the less seed, by different for the stem injection with the microsyringe methods. You can see the image. Here you can see the image of our methodology. You can see the flower injection method, also stem injection methods. And here we can see the results, GH3, IA, and A, different types of results. Here we got the IA, very good result. We got the best result in IA. Then we can see the the control seedless productions okra in IA, 20%, 92%, 95%. This is also IA 100%. This is the with new patent. Already published this work. Seedless student thesis completed. This is the control literature per Ford with pot size, total seed number reduced. The result we can see here. Control is 65, total seed area, seed number 0, seed number 5, seed number 7. We reduce the seed with seedless productions. You can see a nutrient contents, potassium, phosphorus, calcium, magnesium, sodium, zinc, copper. So we can see here we got the good result in different concentrations of Ga3. Then it is, we have seen here that better result. Then control. On the flower project, we have different types of flower. From this flower, you can see this is belong to plant biotechnology as well as industrial biotechnology. We can produce the pigment, natural pigment, organic pigment. These pigments can be used for the textile productions, paper coloring, paper industries. Pigments. These pigments is most important. This belongs to also industrial biotechnology. Then we test the color, chlorophyll test, different types of color. See here, different types of chlorophyll fluorescence interest, and then flower weights and flower size. You can see here, different types of the productions, the flower size higher. In the control, then the size, enlargement, and longevity of hibiscus flower. Only published this work. So this is the thesis, student thesis. Assistant thesis with my supervised many student. Then this is the flower. Induction by kinetic, cytokinin, different type of cytokinin. We induce the flower, more flower. Leaves. Then control. This is the control. This is this is control. This is the different types of treatments. Positive results. Then extra examiner for PhD student. There was extra examiner. PhD student. For PhD student. Then for India and also Malaysia. Then other medicinal plants. Enlargement of rhizomes in Chinese ginger. Chinese ginger by different shade stress and regulators. We use the shade stress and regulators control. These are the control, and then the cheese clots, different type of clots. And then we use abscisic acid, and here we use the black clots. So the white clots and black clots. Black clots is more restricted or prevented the growth. And also this one is very high ABA. We reduce the plant size and we reduce the plant size by ABA hormone, but then rhizome size was bigger. The rhizome size was bigger. So this also these two, we got the result. See also white chest growth and ABA growth regulators size is bigger than the others. Roots also bigger than roots, less roots. Size, fruit size is higher, rhizome size is higher than control and the black clots in case of white seeds and ABM regulators. There is slow production by expansion of roots. My PhD student did this one. tDNA, xylem transposition by IBM, tDNA injection, legume plants, direct tDNA injection, legume plants. This is got the result. This is the technique of transgenic plant production techniques. You can see here tDNA mechanisms. Leaves, oxygen, and cytoplasm opens. These are tDNA regions. This is the TA plasmids, virulence regions, and then opens catabolized origin of the replications. This is called the plasmid with tDNA regions. The transfer of DNA or tDNA or tRNA is the transferred DNA or RNA of the tumor-inducing DI plasmid, directed DI plasmid, tumor-inducing plasmid of some species of bacteria, some agrobacteriums. We read this by agrobacteriums, Trimophaceans and rhizogenans. Two bacteriums. So we can see here different types of. So this is almost all we discussed. This is plant biotechnology, belongs to plant biotechnology and environmental biotechnology. Now we can discuss this is some environmental biotechnology, production of biofuels and the emissions test from waste materials. We collect the waste and produce the energy. This is the now new topic also all of the world. You can see here production of biofuels from waste management, from waste dates. And here we can see the use of renewable energy is a better chance. The global warming issue is caused by using excessive fossil fuels. Renewable clean energy and the resourceful are required for the sharing with fossil fuels to reduce the carbon dioxide. Then this one nitrous oxide, carbon monoxide emissions. So this is the most important for global warming. Common agricultural feedstocks, waste such as waste of sugarcanes, potatoes, pineapples, oil crops, algae, and maize can be used to produce the biofuels, bioethanol, biodiesels. The types of bioethanol has been increased from 3 to 7% to 5 to 4% into the 7 to 8% into the 9. We did this research, and you can see here different types of the fruits research, date farm fruits. You can see here research background global warming polluted by the emitting smokes from vehicles, different types of vehicles produce the pollutions and coughings also of the peoples, polluted by burst smoke, polluted environmental polluted by the smokes from ships, also environmental polluted by the car smokes. You can see here different types of smoke. Smokes are exhausted by car emissions, Nepal, Malaysia, and also polluted in other countries. You can see here polluted environment. Polluted environment movements very difficult. So you see here different types of environment is polluted by excessive vehicular emissions. Here we can see the image, different types of smokes, very difficult to move. Greenhouse gases increased by vehicle emissions. By these emissions, greenhouse gases increases carbon dioxide, carbon monoxide, nitrous oxide, and hydrocarbon. So greenhouse gases increases and affects human health, causes inhaling problem. Lot of smokes, and you can see here cause the inhaling problem in different countries. So this is very dangerous now. So we have to pollute the environment. We have to save our environment. And how to save the environment? We can produce alternative oils. This called the alternative fuels like biofuels. Air pollutions and global warming. You can see here different types of air pollution in different countries, and global warming happens. Air toxic emissions from on-road mobile sources, different types of energy. You can see here in 2009 USA they did different heavy dirty diesel vehicles 10%, gasoline vehicles 14%, others gases 2%, light duty gasoline cars 30%, and also others 44%, trucks and others 4%. So global 1 to 9. So this is the data that now in many countries they're using now biofuels. You can see types of biofuels: wood fuels, rubbish, alcohol fuels, crops, landfills, different types of the sources feedstock, different types of feedstock, leaf biomasses, sawdust biomasses, corn stalk biomasses, corn biomasses, different types of biomasses. You can see around us. Then here we can see different types of from biomass how we can use the energy, methanol, hydrogen, FTDC, electricity productions. This is the sources biomass, and then by different biochemical processes we can produce the gases, and finally we can produce electricity, bioelectricity, biopower from this. This is biodiesel gasoline station. Wisconsin biofuels filling station in Uday 2009. Uday 2009. So this is different types of we can see a biodiesel service stations. Different biodiesel in many countries now established. Also, many scientists are doing this. Now biomass, bioproducts, biofuels, bioproduct, biopower, and different. This is the techniques we have done defense, and we produce also bioethanol. This is one scientist he produced in Australia. So you can see biofuel cell to generate electricity. Biofuel cell generate electricity. And bioenergy flow chart. Bioenergy flow chart also by electricity heats and landfill gas, pipe gas, how we can produce the anaerobic digestions. And then we can see here the electricity heats and the electricity and heat combustions. And this is the energy production, energy crops, and the matter of wood residue from the combustion, upper combustion, is the energy production. He also used the energy production. Electricity production from biofuels. Also, now biodiesel is in the train running. So here are different types of solid waste and recycling materials. Different types of materials. Waste, semi-solid waste, municipal waste, kitchen waste, solid waste, different types of waste we can see here. So this is very important. Lot of waste now around us. We have to control this. Other our drainage system will be blocked. Drainage and sewage system will be blocked. So this is important. We have to use this waste to asset. We can convert it to asset. Now we can see here paper waste, then different types of waste, lots of waste, other waste, different types of waste. This is pedal reactor in India. They are doing this paper waste. So this is different types of biomaterials and biopower. Producing the biopower. You can see here biofuels cell, microfuel fuel cell to generate electricity. How fuel cell? This is the waste, different types of waste productions from fruits, then vegetables also. Plants grown, the plant waste fermentation. And then you can see here different types of fermented. Then after fermentations, here you can see this is called the anode and cathode. Anode and cathode. Producing this is power sources, and this is called the membrane, ion exchange membrane. This is the power sources. And then hydrogen is clean fuel. Then you see that hydrogen plus, and we collected here, and then different under power electric join with photons and forms hydrogen. Yes. Then what is the power from here and here. So this is the power system, how we can do it here also. You can see the different types of we collected the biomass from plant sources. Waste management, recycling process, saving environment from pollution or warming, economically benefits. This is the topics. Or then bioenergy flow chart. Hussain et al. 2007, we introduced this chart, this energy flow chart. This from the plants, sunlight, photosynthesis, sunlight. Then primary producer, secondary producer, consumer, plant crops, fruits and vegetables, biomass. Then secondary consumer, elements, fats, biomass, vegetables, kitchen garbage, elements, and fats, biomass. Then oil, waste oil and biomass. After that it was biodiesel, bioethanol, then biofuel and electricity productions. Then used by the vehicles after heat energy. Then engine start the engine run. So this is called the bioenergy flow chart. Then This is, you can see here, energy flow chart. Here also you can see carbon dioxide reduction flow chart of biodiesel and biotechnology used algae. Different types of algae we used, and then you can see here different types of flow chart. Some algae, carbon dioxide, and then this algae we follow the oil extractions, and then 14 residues, and then biodiesel productions. We follow the biodiesel. What is biodiesel engine? Biodiesel engine can be run. So this is the good topics from biodiesel. The different types of waste: orange skin, tea waste, fruit skin, rotten rice, fish waste, byproducts, onion skin, chili skin, different types of skin. This is not the byproducts. We can use this and then can produce. We collect it and then use for the bioethanol and biodiesel productions. These are types of okay, and then this is the different types of the different countries, Norway and also other countries. They produce now biofuels. 5% they use the bioethanol. This is the dates, seeds, biomass from dates and from fruits, different types of fruits, and now different types of the bioethanol productions. In Germany they are producing from the waste productions. You can see the biofuels. So we collected the also kitchen waste oil, kitchen oil waste oil from after cooking. There is oil waste oil. We collected samples from the University of Malaya restaurant, and then frying oil was made filtration, mixing oil, methanol, and then catalyzed coming after different types of solvents, production hydroxide, versus stirring, mixing oil with alcohol and catalyzed, and then transesterification process. This process called triglyceride, and then catalyst, and then glyceride and methyl ester biodiesel. This is called after the reaction. It was kept overnight, was settling down, separation of biodiesel and glycerin with methyl ester, dry washing by magnesium carbonate, and then drying it in the incubator for 1 hour and storage. So this is we produce in the laboratory. This is biodiesel in Malaysia. So this is the results. We got the good results from kitchen oil, waste oil. Usually after cooking we have thrown this oil, so we can save this oil and we can use this oil and produce the energy. So this is the result also. Different types of viscosity test, different also total acid test. It's all done by standard. Then here we produce the lower layer, glyceride, upper layer, biodiesel productions. In terms of biodiesel production, you can see here this produce the biodiesels. Cooking oil, biodiesels. This is the LP sample collection. One project. This is my PhD and master student. We work together in the field for to control the environmental pollutions in one place in Malaysia. This is the we collected the algae, and then in the laboratory we did the laboratory work on food, produce the oil extraction, algae separations, biomass and transesterification, shaking process, and then we produce the biodiesel. This also waste collection, kitchen rubbish, shrimp byproducts we collected. And this is the medicinal plant collection we collected, and garbage collected here. Different types of waste fruits also we collected, and then different waste fruits we collected from other cities. From then bioethanol productions from rotten fruits, different types of rich fruits. We produced the different types of seed are separated and yeast separation, different was added to different concentrations. Sample was kept in incubator, and the fermentations process, glucose yeast plus yeast ethyl alcohol, and then we put carbon dioxide. Then we open it to a small bottle, and then after fermented methanol was filtered and separated from biomass. And also there was ethanol and water. Then we separated the water bioethanol from water by this water vacuum This is called the evaporator. Evaporator. Then this is the different types of also samples, water samples, fruit samples, fruit waste samples. You can see here different types of samples were produced, and in the laboratory we worked. We did a lot of works. You can see here the results. Got the positive results by thermal production or energy production. PS glucose test, different types of STM standard and EN standard, European norm, American standard for testing and materials. According to their standard, our result was very good. Ethanol from the dates, and also yeast with the yeast cellulose amylase. And then we see the SLI was 4.5 gram, 1 gram. There is no difference, but little bit better in higher 0.5 grams. So here also because of seeds. Then we can see fuel consumptions. We with the engine, we use the fuels, different types of engine. This is bioethanol productions. Then we electricity generation or waste biofuel using fuel cell, different fuel cell. And then we use the bioenergy cell, and then we see that by this produced energy, bioethanol engine start means the fan and the others device started. So this is the bioelectricity generations testing in fuel cells machines. So this is the my understanding. Many I supervise many undergraduate, and this is the thesis, and this is the this books, and then my books, published books, and many theses and papers, publications. This is one student, my master student, she collected this chicken waste and fish waste, and the produced the energy from this. He produced the biodiesel. Then the incubator heat, and then produced the there was the oil, fat. After that, this fat can be used for the biodiesel productions. And here also different types of the waste materials he collected. We also collected the fruit waste, and then visit the waste place and the advice how to arrange waste. I advised the people, and we visited many places and advised the people how to clean the environment and how to control the pollution of the environment. Then published books, and then this the topics, different types of the then other industry bioplastic preparations or biomaterials from the pills. We can see here cellulose productions, different types of cellulose productions we did. And then here we can see car bumper from dates and olive waste, different types of waste in the Middle East, Saudi Arabia. This is the cellulose, plants lignocellulose. By the lignocellulose we can produce different types of plastics, bioplastics and bioplastic materials. And we have different types of materials within the process of bioplastic car bumper with the acid hydrolysis, nanoparticles produced. Yeah, in short measurement, you can see here methods: acid hydrolysis, pyrolysis, and combustion. Nano-size particle by acid hydrolysis, and there are also measurements, different types of measurement. And then heating and the plastic preparation, not types of heating and plastic preparations. These are the topics. You can see here drying and testing, different types of color test, and then drying of different types of plastics from this type of materials. This type of materials, horn materials, and banana peel materials. So then they put the sweating drying the plastics, and different types of bumper shaped, the bumper shaped, car bumper shaped, like the this shape, car bumper shaped. This is possible. Actually, we made this as a laboratory scale. Then when industrial people, they will do it as commercially, and then as commercial purpose, then they would it will be very good, and they will see the feasibility, how it can be possible. Then we shall consult with them, and at the time they need us to make consultations, to make the advice to them. That's why it is important we publish this paper. He also tested the absorption test. We did the absorption test, many absorption tests, and we did the formatting for molding of the bumper shape, and then this lignocellulose. From this lignocellulose also we can produce the different dashboard, different types of plastic, and the cars, other materials, car bumper process industry. This is the useful for how we can do this. This we can see the nozzles. This is the balance. This is the molded plastics. Just here, put here, and the raw parts, and the raw plastic is good for driven here. Then this is the paste materials. This paste material we can produce from the waste. This is the main topics. Then after that, this formatting others. This is industrial people will do this. The plastic screw for bone joint. This is different types of screw for the medical use, biomedical use. Then X-ray, we produce X-ray, X-ray film. And then nano coating of the tablet coating. So we can produce by this way. So there's some editorial members. I am some editorial members of many journals, biotechnology, physiology, and others. Issues of plant science. And then also I'm associate editor and editor of the different journal. This is the honor by rector, University of Haile. I got the rector award. This is the honor award and recognition of my world class as a world class scientist. They put my name in 2011. Okay, and then this is PhD thesis checking and all thesis together. I supervised a lot of students, PhD, masters, and undergraduates. So this is all about. Just at a glance, I published a lot of books 15, and then books chapter monographs 26, total published 175, including book 210 publications, and then ISA publications, and also the Scopus total more than 110, ISA publication 55, presentation 52, ISA index citation 2430. So this is all about achievement. I completed many research project. This is here, and then here are some lab image we worked in Malaysia, some different laboratory work, and then here you can see the student work. And here I attended the conference in Sudan. I attended in the Egypt, and here Lebanon, and many countries, and then this is my publication. So, some of my thank you very much for listening, watching, and subscribing my channel. Thanks again.